Good afternoon, Dr. Cormac.
Hello.
Hi, I'm
Dr.
Marwa.
Marwa.
Hi,
Hi,
how are
Dr.
you?
Marwa. How are you doing?
You put on the video. This is good. To interact better, to see
Oh,
the
yes. Yeah, I
students.
think so. Yeah, I think so. Otherwise, it could be a recording.
Okay, we are waiting that the rest of the students
Sure.
to join the meeting because their average, I think, 100 students or something like that. Because we are joining two groups. I will
Okay.
speak in Arabic to address the students, and then I will
Sure thing.
just let
I won't speak in Arabic.
Okay.
Except at the beginning.
مساء الخير يا دكاترة. انتوا سامعيني طبعا. انا دكتورة مروة تمام معاكم من الكلية. حتى raise your hands عشان you are muted in order ان انا يعني انتوا سامعيننا. دكتور كورماك هو اللي هيديكوا اول 5 اسابيع. معايا سامعيني ولا الصوت مش واضح؟ اوكي. هو هيديكوا اول 5 اسابيع، هيديكوا جزء مهم جدا هو الجيناتيك انجينيرنج، وبعد كده انا هكمل باقي معاكم باقي الكورس. فيا ريت يعني to listen carefully and to be attentive to the doctor عشان هو اللي هيديكوا الجزء ده. It's very interesting part and then he might ask your interaction. فبليز يا جماعة أنا بكلمكم بالعربي يعني to interact مع الدكتور، وأنا هبقى joining لو أنتوا عندكم أي حاجة، لو مش فاهمين أي حاجة you can ask during the lecture whatever you want. Okay. احنا you're joining and we have now 23 students participants. I will wait Dr. Cormac. I will wait more 2 minutes
Sure.
to in order to for more students to join. Okay.
Sure thing.
Thank you.
Will I share my screen now or
Yes,
share
you
the
can
file?
start sharing your screen and preparing everything and till the rest join.
Okay. This is it here. You can see that? Yeah.
Dr. Cormac, you can start. I think we have almost 50 students.
Okay.
You can start to introduce yourself and then you start the lecture.
Okay.
I'll be with you for any assistance or anything. I'm online. Okay.
Okay.
Thank you,
Thank you.
Doctor.
And will I see the comments automatically?
And you can see the comments in the chat. They send you asking questions. You will find down you have security, participants, and chat. At the
Okay.
chat, while you are presenting your lecture, you might see that they are, for example, increasing number of chats, therefore they are asking questions.
Okay. I'll just,
Okay.
hopefully I'll see that.
Take your time. It's the first time. Take your time. You can every now and then you can go on through the lecture, and because you are going to you are sharing your screen, and then the chats will appear يعني down.
Okay, they should
Okay.
appear automatically. Okay, thanks very
Okay.
much. Okay, thank you, Dr. Marwa.
Thank you. يا جماعة بصوا اللي هيكتب أي سؤال من فضلكم بالإنجلش طبعا عشان يقدر يرد عليك. شكرا.
Shukran. Okay, thank you everyone, and assalamu alaikum to everybody, and tasharafna. Pleased to meet you. I had to translate myself. So I am Professor Cormac Gahan. I'm from the School of Pharmacy in, hopefully you can see my pointer there, in University College Cork, and I'm going to talk to you today about regulation of gene expression and also the history of biotechnology. So just to give you an introduction to myself, because it's interesting to see where people come from. I am from Cork, which is in the south of Ireland, which is near England. This is England here on the right-hand side, Ireland, and on the western side of Europe. And we have a lovely campus, nice campus, old university campus here. And I am present in this building here, and also in this building here, but my labs are in this building here. So we have my own research is actually in the area of microbiome science, so the bacteria that live in the gut. And I'm part of a research center called the Microbiome Center. And we are very interested, this is by way of an introduction, this isn't part of the lecture, it's just to show you what my research is. I'm interested in these gut bacteria and how they influence the host. And specifically, I direct you to, we published a paper a few years ago, which might be of interest to pharmacy students, because we realized that all of these gut bacteria that live in our guts can have a lot of metabolic activity, and this metabolic activity may interfere with or indeed increase drug activity. And so we wrote a paper about 4 years ago, which is free. And I would just suggest that if you're interested in how bacteria interact with drugs, and you can look at this. You can look at this paper. It's free to download, and it gives you an overview of how these gut bacteria may influence pharmacy and drug availability in the gut, and so on. It's not related to today's lecture. You don't have to study it. It's just to show you the type of research work that we are doing in my lab in UCC, and I think it's a very interesting study in the future. And these are some of these are not people in my lab, but they're people in the institute that I work in, and that is just really by way of an introduction to you, so that you can see the area that I'm interested in and the type of work that we do in Cork, which is very much focused on these gut bacteria and how they influence our host metabolism and drug metabolism and so on. So we've got a big, large research institute in that area. Okay, to the lecture. So the actual lecture itself today. For the first part of the lecture, I am going to be talking for probably about half an hour, maybe a bit more than half an hour, on the history of biotechnology and genetic engineering. So what is the history of where the industry, the pharmaceutical industry, has come from? Around the 1970s, there was a major revolution, if you like, in the biotech industry. And so I'm going to talk about that from a historical point of view, but also there's quite a bit of science in this part of the lecture. And then in the second part of the lecture, probably the last 20 minutes of the lecture, and then the remainder of next week's lecture, we're going to talk a bit about gene regulation. So in that part of the lecture, we're going to go from biotech industry into a revision of transcription and translation. I realize you know this biology about protein synthesis. We're going to revise this and show you where gene regulation can be of benefit to the cell. So we can regulate transcription, and this can be beneficial to the cell. save energy. And then we're going to talk about this gene regulation, why it occurs and how it happens. We're going to look at the operon model, the fact that lots of genes can be regulated at the same time. And then we're going to look at how these operons are induced or repressed, and an example of each type of operon, the lac operon and the ARG operon, arginine biosynthesis. So a lot of this will be next week. And to tell you the truth, next week's lecture is probably more detail. There's probably more detail. This week's lecture, today's lecture, is a bit more gentle. It's a bit easier to follow, a bit more straightforward. And then next week we get into a bit more molecular detail. So the introduction to genetic engineering and modern biotechnology, this all happened between 1968 approximately and 1980. Okay, so as you know, the DNA molecule was first appreciated. The structure of DNA was first appreciated in 1953 by Watson and Crick. So we understand the structure of the DNA molecule. But how can we exploit this? How can we use this technology to make new drugs and to advance the pharmaceutical industry? Okay, so this is what happened in between 1968 and 1980. And before 1980, drugs were produced by either chemical synthesis or they were isolated from nature. So you could chemically synthesize a drug, including an antibiotic, could be chemically synthesized by chemical synthesis, or it could be isolated from a bacterium or a fungus. You could isolate the drug, okay? And the same is true of insulin. So for insulin, we could actually isolate that from the pancreas of cows. So you could isolate insulin from the pancreas of cows. But what modern biotechnology allowed us to do is to make insulin by genetically engineering E. coli and to actually make this biologically active drug, okay? Much better, much cleaner, as we'll see. So what I want to show you in this lecture is that this is a much cleaner operation. It's much more precise, and it is a major advance that occurred only in 1980, okay? So not so long ago, really. And all of these discoveries were made in the west coast of the United States in a very small area near San Francisco. So this is in the western coast of the United States where all these discoveries were made, either in San Francisco or in Stanford, which is in Palo Alto here. So Palo Alto, Stanford, or San Francisco or Berkeley, okay? So we're going to go through the history of this, a major, major advance in the pharmaceutical industry. And it goes back to around 1968 when this scientist called Stanley Cohen was in Stanford University, and he actually still works in Stanford University. So I've actually visited Stanford, and I know people who've worked with Stanley Cohen. He still has a lab there. And what he found was he was one of the first people to discover the presence of plasmids in bacteria. So he found that bacteria, yes, they have a chromosome, which is the main source of DNA, but they also have plasmids, which have another source of DNA in the cell. Okay, so this is a smaller, replicating, self-replicating piece of DNA. And actually, this diagram is probably not very accurate because actually there are many copies of plasmids in the cell, maybe up to 50 or even more copies of the plasmid DNA, where there's only one copy of the bacterial chromosome. So these are present in multiple copies, and they can often contain the genes for antibiotic resistance. Okay, and here's a typical plasmid up here, which shows you the structure of the plasmid. It's a circle, but it also has the gene for ampicillin resistance on this plasmid. Okay. Now, what he found is that he could transfer antibiotic resistance from one bacterium to another through the transfer of plasmids. So in other words, he found that these plasmids could be transferred from one bacterium to another. And this was a very important first step in molecular cloning of DNA. This was around the same time Herb Boyer, who is an interesting character who we'll see throughout this the history of biotech. He was working in San Francisco University, which is about 20 or 30 kilometers away from Stanford. And he was working on restriction enzymes. So these restriction enzymes are molecules that can chop up DNA at certain restriction sequences. Now, he wasn't there, he wasn't actually the first person to discover them, but he was using them in his lab he was using them in his lab to do experiments in E. coli. And these restriction enzymes, what they do is they cut DNA at a very specific point. So you can see here that you have EcoR1, which is a particular type of restriction enzyme that comes from E. coli. So ECO stands for E. coli, and R1 is the strain of E. coli. So this particular restriction enzyme cuts DNA at only this site, GAATTC, cuts at this specific point to create these sticky ends to the DNA molecule. So we'll see in a separate slide in a moment, we will see how this can be used to clone DNA in E. coli. Okay, but he was an important character in this story. And it was work that was performed again in Stanford University by Paul Berg, another scientist working in the area. He was the first to put these two ideas together. He said, well, what if we chop up a plasmid, up, and then we chop up some DNA. We use the same restriction enzyme to chop them up, and then we use an enzyme called DNA ligase, which ligates them together. Okay, so I'm going to look at this. I have another slide where we look at this process. But the important point is that if you put these two ideas together, plasmid DNA and then a chopped up piece of DNA, we can actually put this chopped up DNA into the plasmid. You see it's gone into the plasmid here. And then we can clone this into E. coli. We can transform E. coli with this DNA. And actually, he was able to select for ampicillin-resistant colonies because there's an ampicillin resistance gene on the plasmid. So that means that so if you put ampicillin into the agar plate, only the E. coli cells that have received the plasmid will grow on the agar plate. You're selecting for ampicillin resistance. Only the cells that have received the plasmid, the ampicillin resistance plasmid, will be able to grow. You know that all of these colonies, they have the plasmid. Okay, so he was the first to perform these very quick DNA cloning experiments. And it was only a year later then that Herb Boyer and Stanley Cohen got together and they performed what I think is probably one of the most key experiments in all of biotechnology. And that was performed in 1973. They took some of the DNA from a toad. So if we go up to the example up here, this DNA came from a toad, whereas the plasmid obviously came from a bacterium, and they were able to clone the DNA from a toad into this bacterial plasmid. And the amazing thing was they were able to put that into E. coli, and they were able to make the E. coli produce proteins from the toad. Okay, so this seems very routine to us now. We do these experiments, we can do this type of experiment very easily now. But back in 1973, what it showed them was that the DNA from a toad is very similar to the DNA from a bacterium. We can get, it's so similar that we can actually get a bacterium to produce and replicate DNA from a toad and get it to make toad protein. Okay, so this was an incredible experiment that showed that molecular cloning from one species, from the toad, into another species, the bacterium, that this is possible. Okay, so it is possible to make eukaryotic proteins in a prokaryote. And they suggested, well, it should be possible to make human proteins in an E. coli strain. Okay, and that's exactly what they did next. So this was really important, showing that the species DNA was interchangeable and that you could do this type of molecular cloning experiment. So what happened was Herb Boyer decided, well, I'm going to go and we're going to make a company. We're going to develop a company and we're going to float this company on the stock market. And this company is going to take this technology. The company is going to take this type of technology that they're doing here, and we're going to start to make biologically active, important drugs using this technology. Okay, and that's exactly what they did. He was able to become a partner with Bob Swanson, who's this guy here. Bob Swanson had the money. He was a venture capitalist who could put money into this, and Herb Boyer was the scientist. Okay, so you've got a scientist and a money person, and if you put them together, you can create a company. So they created a company, and the company was called Genentech, for Genetic Engineering Technology. Very early stages of biotech, 1976, to produce biopharmaceuticals. Now, as you all know, biopharmaceuticals, we use them all the time. Today, nearly all the drugs we make are biological drugs, which are biopharmaceuticals. Well, a large proportion of them are, and this is the modern biotech industry. Okay, so it started in 1976 from Genentech. We'll see how Genentech developed the technology for insulin. That was their first drug, and they went public. In other words, they offered their shares on the stock market, and they made $35 million in an afternoon, which was a lot of money. Still is a lot of money, I appreciate, but I suppose it's not quite at the billions of euro and billions of dollars that we see for Google and so on nowadays, but it was a lot of money at the time. So it showed the industry was about to change by this process into genetically engineered drugs. And they were able to clone human insulin into E. coli, and they won the race, if you like, to clone human insulin, and they were able to license this technology to Eli Lilly, actually, and it was Eli Lilly who started making the drug. In 1982, human insulin was the first recombinant DNA drug that was approved for human use by the FDA. So that's not very long ago. Okay, so that's why this technology is important. This history lesson is important. Okay, but the most important thing about this history lesson is this. Look on the left-hand side. These are the old production procedures for insulin. You could take, this is a cow pancreas from a cow. You would extract the insulin from the cow pancreas. So these are lots of biomass in the cattle industry. They mash them up, and they used chemical processes to extract insulin. And then what you're doing is you're injecting a human with insulin from a cow to control their diabetes. Okay, so this was useful, but the humans would often get infections. They would often have allergic reactions to the insulin. The insulin sometimes wouldn't work very well because it's cow insulin. It's different to human insulin. So there are a lot of downsides. People would often die of adverse reactions, and this is not a good system to use. Whereas now, look at this clean system. In this vat here, you have E. coli producing insulin by genetic engineering. Much cleaner system. We can isolate that insulin, and of course, it is human insulin, making human insulin. It's not cow insulin. It's human insulin as a drug, which can be produced as a sterile formulation, and this is what is injected into the diabetic patient. Okay, life-saving. Now, of course, there's many thousands of drugs that are now produced by this. Interferons, different drugs are produced by this using this technology. So how do you do it? How do you actually clone something into E. coli? And actually, Owen Fleming is going to go into a bit more detail on this in a few weeks' time. Owen Fleming from Cork is also going to take some lectures on genetic engineering and biotech. But just to give you a very brief overview, how do you clone something into a plasmid in E. coli? Well, you use our friends, the restriction enzymes, to cut both the plasmid DNA and the DNA you want to splice in. The enzymes will cut at a specific site. So the nature of, say, EcoR1 is that it cuts at a specific restriction site. So this is the DNA molecule. You've got a site here, GAA-TTC, GAA-TTC. And the amazing thing about this is that it actually reads the same forwards as it does backwards. Okay, so the cut site, it's called a palindrome. The cut site here is GAA-TTC, and you see on the other strand, the lagging strand, GAA-TTC. So it reads the same backwards here. So this is a very clever enzyme. It can recognize the sequence, and it cuts here at the G, and it also cuts here at the other G on the complementary strand. And what this does is it creates these sticky overhangs. So here you've got, imagine if you cut it here, you've got G on one side, and then you've got the TTAA underhang here like that. Okay, and you've got it on the piece that's been cut out, you have an overhang. So if you cut this green DNA, this could be from the toad, this could be toad DNA. You cut the toad DNA with EcoR1, it cuts in the exact same spot. Okay, an AATTC overhang. If you cut the plasmid from the E. coli, this is from the bacterium in blue, you'll see that this here, this overhang will neatly align to this. The AA will bind to the TT, and the TT will bind to the AA here. So we can discard these. This will come over here, and here you can see that these will align. Okay, so that's the beauty about genetic cloning. We can use these restriction enzymes to produce complementary overhangs, which will bind to the cut DNA. So in other words, if we cut DNA with the same enzyme, you can actually ligate that DNA together like this. And this is just using, you can see base pairing of DNA to each other. The T will bind to the A, and here the T here will bind to that A. Okay, and then you can use an enzyme called ligase, which ligates all that together. Okay, so this is the basic technology for cutting with an enzyme and then ligating together. And this is I won't go into this. This is the exact same slide showing that yes, we can ligate these together if they're cut with the exact same EcoR1 enzyme. Luckily, we have lots of different enzymes at our disposal because these are enzymes that are produced by a lot of different bacteria, and actually we can, there's a lot. Once those of you who may go into some research will see this in the lab, working in the lab. And so we can take a cloning vector, which is like this one here. This is a plasmid. It's a plasmid, which is a circular piece of DNA that we explained at the beginning. And this particular plasmid is PBR322. It's a typical plasmid that we sometimes use in the lab. But what you'll see when you look at a map of the plasmid, you'll see a number of interesting features. One is that here there's an EcoR1 site, which is a unique site. So if we cut this plasmid with the enzyme EcoR1, we can open it up. It opens up. There's only one EcoR1 site. It's a unique site. So all this will do is it'll open it up. It'll linearize it into one piece of DNA. Okay, it's not a plasmid. It's not circular anymore if we cut it with EcoR1. The other interesting feature is this ampicillin resistance gene. And this means that any bacteria that has this plasmid will be resistant to ampicillin. Okay, so it'll grow on a plate, an agar plate with ampicillin on the plate. Okay, so they're the main things about this plasmid. We can cut it using EcoR1 in one site, and we can use that to clone in pieces of DNA. And then we can select for bacteria that have received the plasmid by selecting on ampicillin resistance plates. And that's exactly what was done in this from a previous slide. We can cut the DNA with EcoR1. We can cut the plasmid with EcoR1. We can ligate the DNA into the plasmid and then select, and then we transform it into E. coli, and then we can select for only those E. coli that have received the plasmid by growing them on ampicillin plates. All the other E. coli that are in the mixture that don't receive the plasmid, they're killed by the ampicillin. Okay, so we select for recipients on ampicillin plates. Okay, and then we can actually visualize this process by using a DNA gel. Sometimes you'll be watching TV programs like these programs on TV, and they'll be looking at, maybe not using a microscope or a magnifying glass, but they'll be looking at these gels to see the DNA, to visualize the DNA. Now, you may have used these gels. I don't know. Maybe you've used these already in different labs to look at DNA. But the gel itself will run. What it does is it's a system for running DNA by size fractionation using an agarose gel. So you've got an agarose gel base, and then you load the plasmid or the DNA into this like this. This is just so that we can see what we're doing in the lab. And then by applying an electrochemical gradient, an electric gradient, we can run this so that we've got a power pack here, which is applying a gradient, and it's running from the negative to the positive electrode. In other words, we're setting up an electronic charge here through the system. And that, because DNA is negatively charged, it will actually run towards the positive electrode. Okay, and it migrates then according to size, with the smaller molecules migrating faster through the pores. So the tiny pieces of DNA will run very quickly towards the positive electrode, and the larger ones will become trapped higher in the gel. Okay. We can add a thing called ethidium bromide, but there are also other dyes which have replaced ethidium bromide. We can use Cyber. There's other dyes which can bind to DNA and make it fluorescent under UV light. Okay, so some of what I'm going to say becomes clear now when we look at this. Here is a typical gel here, and in the gel we have run our different pieces of DNA from the negative electrode down to the positive, and so the smaller pieces of DNA have run furthest. This is a small piece of DNA, 1,800 base pairs. It's run all the way down here. The larger pieces of DNA become trapped at the top of the gel. So they become trapped here or up here in the gel. Okay, this is where the larger pieces become trapped. Okay, so by running the DNA, we can see it. Okay, so this is, the reason I wanted to show you this is these are the practical aspects of doing these experiments. So what we have at the top is, this is the piece of DNA that we want to clone. So this might be the gene for insulin. Okay, we want to clone the insulin gene into a plasmid. Okay, and this piece of DNA is the insulin gene, and it runs here in lane B to around 4,000 base pairs. Okay, so you got a 4,000 base pair gene, big gene. It's been cloned into this plasmid. Okay, and it's cloned in because you've got these EcoR1 sites which allow this to stick to that. Okay, and here's the plasmid cut on its own. It's in lane C. This is 5,000 base pairs. Okay, so you've got a 5,000 base pair plasmid. So on its own, this is what the plasmid looks like. It's cut at one site, it opens up, it linearizes, and there you have the plasmid. The insert, the insulin gene, is here. Okay. So what happens when you put these together? You ligate that one to that one. There might be a chat. Yeah, so you can ask any questions. I know I'm maybe going a bit quickly, but anybody can ask a question. You can ask on the chat, or if you want, you can ask Professor Marwa, and she could translate if you wish. Please don't ask me in Arabic. So anyway, maybe it is clear to you. So this is the, whoops, go back. Oh, here we go. So the plasmid looks like that. It's opened up. The insert that we want to clone into the plasmid, this is the insulin gene, looks like this. What happens when we stick them together? Well, we get a bigger plasmid here, like that. Okay, where you have the insert, which is the insulin gene, ligated into the plasmid itself. Okay, and of course, when you run this, this is around 9,000. Okay, because 5,000 plus 4,000 is 9,000. So we can see on the gel that this is a big plasmid now. It has the insulin gene. It's running to around 9,000 base pairs. Okay. So that's just a very basic example. It's simplified a bit, but it's a basic example of how we can do these experiments in the lab. We do these experiments all the time, clone things in the lab, express them in E. coli, and of course this plasmid can be expressed in E. coli, and now it'll produce insulin. Okay. Now, just certain technologies that are important, and these were developed along in the 1970s, around the same time. And this is to actually tell you how insulin was cloned. I want to tell you a bit about some of the technologies that they used. Okay, of course they used this basic genetic engineering technology, but they also used a number of different technologies. And what they did at Genentech was that they used a technology called synthetic DNA, or DNA synthesis. And in this case, what they did was they built the gene using a silica gel support. We won't go into the details of this, but there's a technology called synthetic DNA, or DNA synthesis, where you can add the nucleotides individually, and they will attach and form DNA by a chemical method. Okay, so we won't get into the detail, but the important thing is you can create DNA using a machine by chemical synthesis and by just feeding the machine the correct nucleotides. So,
Dr. Cormac,
yeah,
sorry
sure,
for
sure,
interruption.
sure.
I will leave you in this the last half an hour, and then I will join you again in the next with the next group at 4:30.
Sure
Because
thing.
I have
Okay.
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 Thank you.
Thanks, Dr. Mara. Thank you.
You're most welcome.
Okay. Yeah. So if anybody has any questions, please let me know. I can go to the, hopefully I can go to the screen chat. I've lost the chat. I've actually lost the chat. So if anybody has a question, maybe you can turn on your microphone and stop me. Oh yeah, sorry, here I am. So I do have the chat if anybody wants to type in chat. So what they did at Genentech was to create the insulin gene using DNA synthesis, which is very clever. So the DNA synthesis allowed them to synthesize the gene for insulin by using this chemical synthesis of insulin. Okay, and the beauty about this is that they didn't have to actually clone the gene from the human. They could actually create the gene in a test tube. Okay, and so this is exactly how they did this at Genentech. And also the beauty is that you can even potentially introduce changes to improve the protein. So you'll see when you go into clinical practice that there are lots of different types of insulin with different properties, and that's because you can actually make mutations in the insulin gene to insert maybe a different type of amino acid at a specific point. We'll get to that in a minute, but that's one of the benefits of this technology. You can actually make long-lasting insulin, or you can make short-acting insulin, whatever you wish, using this technology. Just as an aside, this technology was probably the high point of this technology was in only about 10 years ago, a paper published in Science, where they synthesized a complete genome for a bacterium. Okay, this is a group in the United States. They synthesized the entire genome of a bacterium, and this may have major implications for vaccines, therapies, gene delivery, and so on. So back in 1978 and 1980, Genentech were able to synthesize short genes, but bring us all the way up to the modern age, and we can actually synthesize an entire genome. And they arranged the DNA fragments in yeast. They transplanted them into hollowed out bacterium. So incredible science to make a whole new organism that hadn't been seen before. So this is synthetic bacterium, the first synthetic organism, which improves therapeutics, which has lots of different implications. Okay, so let's get back to insulin. And so the science took place at Genentech in 1978. Before this, we've already seen that insulin was made by harvesting from the pancreas of cows or pigs. The scientists at Genentech, Herb Boyer, had already cloned smaller protein into E. coli. And they said, well, our biggest goal, and what's the most important drug we can think of synthesizing initially, and that was insulin. So they said, well, what do we know about insulin? what do we know about how it's produced in the body? Well, insulin is actually produced as a proinsulin, which is cleaved to remove an intervening peptide to form active insulin. So we'll see this on the next slide. And so if we look at the structure of insulin, we can see that actually in the body, in the human body, it's actually synthesized as this much bigger protein called a proinsulin. Okay, so proinsulin, whoops, sorry. Proinsulin is made as this big, long insulin molecule. And what happens with proinsulin, it's produced in the pancreas as an 86 amino acid protein. What happens is, nothing. And what happens is these two parts of the molecule, what's called the A chain and the B chain, are actually linked by disulfide bonds. So these are linked together by bonds. And then in the body, the proinsulin is cleaved to give active insulin. So this part here, the arginine to the other arginine, this yellow part is cleaved and discarded. That leaves these two pieces joined up, the A chain and the B chain joined up to make active insulin. Okay. Now, the beauty of this is that these are very small proteins. The A chain is only 21 amino acids. Okay, so this is what they were talking about at Genentech back in the 1970s. They said, well, the A chain is only 21 amino acids. That's only 63 base pairs of DNA. You remember from your biology that you've got triplet codes of DNA which encode each amino acid. So you multiply that by 3, 21 amino acids. So therefore you have 63 bases of DNA that go into making that those 21 amino acids because of the triplet codons and DNA coding. And then the B chain is only 30 amino acids, slightly bigger, and that's only 90 bases of DNA. Okay. So what they said was, we can actually synthesize, we can use this process here on this slide, we can use this process of DNA synthesis, and we can synthesize, because they knew the code, they knew the amino acid code, they said we can use DNA synthesis to make 63 bases of DNA for the A, encoding the A chain, and 90 bases of DNA encoding the B chain. Okay, so they said this we can do. This is relatively simple, and that's exactly what they did. They synthesized the genes that encode the A and the B chains, and they actually did this separately. So they said, well, in one E. coli we're going to get that E. coli to make the A chain, another E. coli we're going to get it to make the B chain. Okay, and they did a very clever thing. They ligated the A chain DNA. So here's the DNA for the A chain in a plasmid. They ligated this to beta-gal protein, which is a standard E. coli protein, which actually we're going to look at in the next lecture next week. Okay. So they said, well, let's link the A chain to the beta-gal protein because we know we can trick E. coli into making this. We can get E. coli to make this beta-gal chain, and at the same time, it'll make the insulin A chain. And then in a separate test tube, they said, we'll do the same for the B chain. Okay. As I said, the B chain is slightly bigger, 90 amino acids, 90 base pairs, sorry. So they were able to create these clones, transform them into E. coli. And what they did was they got the E. coli to make, it's a fusion protein, the beta-gal protein is linked to the A chain. So here you can see in pink, the beta-gal protein linked to the A chain of insulin. This could be grown up in culture so that the E. coli makes lots of it. And then what they were able to do is actually, they took a thing called cyanogen bromide, and it's the cyanogen bromide which cuts this here. Okay, I won't go into the detail, but the cyanogen bromide is a chemical which can cut here at a specific point here. Okay, so they're able to cut these are the proteins. They were able to cut the beta-gal protein away from the A chain and release the A chain here. And in this side here, they were able to cut the B chain off like that. And then they simply used a chemical process to recreate these disulfide bonds. And here you have active insulin. Okay. So this seems like a long process, but if you go, what I want you to do is when you revise this, you go back over this process and you'll see that it all makes sense. For insulin, you just need the A chain and you need a B chain, and they need to be tied together with disulfide bonds. So what they said they would do is they would create an E. coli that makes the A chain and create an E. coli that makes the B chain. They can isolate these and then tie them together using a chemical process to make active insulin. Okay, I might have a chat. Okay, so could I explain this slide again? Okay, and what I'll do is I'll go back and I'll explain both slides. Oops. I'll just explain how we get from this to this. Okay. So for this, at the top, this is how the body makes insulin. This is how it generates from so there's a gene that's expressed in the pancreas. It's in the human genome. It makes a very long insulin protein. And the two chains of the protein, this A chain here and this B chain here, they're actually tied together using disulfide bonds. So what happens when insulin is released, it's a thing, and another enzyme comes along and it cuts the proinsulin. This is called proinsulin. It cuts this yellow piece away, and that leaves these two pieces tied together. And this is actually the structure of insulin as it occurs in the body, as it occurs in the bloodstream. So blood-borne insulin looks like this. When it enters the bloodstream, this is what insulin looks like. Okay, it's an A chain tied to a B chain. And so this is what, when we want to make insulin in the lab, this is what we need to make. We don't need to make this here. We need to make this one here. Okay. And so scientists said, well, at Genentech, they said, well, this is only 21 amino acids, and they know what these 21 amino acids are. Okay. So if you know the amino acid structure, you can make the DNA that encodes that amino acid structure. So we know the genetic code. We know that there's a specific code that encodes valine here, and so on. But all you need for 21 amino acids is 63 base pairs DNA. Okay. So then, in other words, once you know the amino acid structure, which they do know, you can create a piece of DNA that encodes that, and the piece of DNA that encodes that is 63 bases. The piece of DNA that encodes the B chain is 90 bases of DNA. Okay. So they said, what we're going to do is we're going to actually synthesize these. We're going to go back to this technology of synthesis. We're going to synthesize both the A chain, the DNA that encodes the A chain, and the DNA that encodes the B chain. And we're going to put this DNA into a plasmid, and we're going to ligate it to a beta-gal gene. This is a gene that encodes this is another enzyme in E. coli called beta-gal. So in other words, this is an E. coli enzyme, whereas they're ligating it to a piece of DNA that encodes the insulin A chain, and then in this case, the B chain. So what they're doing is they're tricking E. coli. This E. coli thinks it's producing the beta-gal gene, the beta-gal protein, but it's actually also producing the alpha chain for insulin. Okay. So this is transformed into E. coli. The E. coli produces the protein that encodes the beta-gal gene, but also tied onto that is the A chain. Okay. You can use a chemical called cyanogen bromide to cleave that. So that cleaves this away. The A chain is now free. And in another Petri dish or another test tube, you can make the B chain, and you can ligate these together using a chemical process. So they use chemistry to make these disulfide bonds. In other words, you can mix the A and B chains together, produce them separately, mix them together, and create these disulfide bonds. Okay. And the beauty about this is that you can produce this now under clean conditions. Okay, so you start with a tube full of E. coli, you can scale it up, you can grow it in these thousands and thousands of liters of E. coli. They produce either the A or the B chain. You can then process and extract. You aseptically fill vials, the finished product. You test for sterility, LPS activity, biological activity. So now you've got a very clean, the take-home message here is this is a very clean system, extremely clean for making these sterile drugs. The other beauty about this is that you can actually, you're not limited to making the natural form of insulin. So the beauty about this system, so humulin is the natural form of insulin, and this humulin is human insulin. However, you can actually make different forms of insulin by changing the amino acid structure. You can change the amino acid structure by changing the gene sequence. Okay, so we can make other types of insulin. Here's a Liprolog, a short-acting insulin. It's got different pharmacokinetics in the body and different half-life in the body. And so this might suit some patients. Some patients might prefer to take short-acting insulin. Other patients may take a longer-lasting insulin. Here's one, Lantus. Okay. So when you go into clinical pharmacy, you'll see these different variants of insulin because they're used in clinical pharmacy all the time. And here's an example, Lispro. So Lispro is on this here. Lispro is this one here, Humalog. Lispro, all Lispro is a switch of the proline and the lysine. Okay, so here in Lispro, instead of a proline here, you have a lysine. Instead of a lysine here, you have a proline. And this is done through genetic engineering. In other words, we can use genetic engineering to change the structure of insulin so that it has different half-life in the patient. Okay, and this is really important because we can improve the outcome for patients by changing it. Okay, so that's the topic of genetic engineering. And but how does this link to the next part of the lecture, gene regulation? So we've got about 5 minutes, and I want to make a 5-minute start on gene regulation and why is it so important? Well, the two links between the two is that we have to understand gene regulation because Genentech fused the insulin proteins to beta-galactosidase gene from the lac operon. We'll see why it's our friend in a minute in next week's lecture. And this was to trick the E. coli into producing the foreign protein. So we could trick E. coli into producing the foreign protein. So the scientists knew exactly how to regulate this beta-gal gene and produce large amounts of the recombinant protein by regulating the lac operon. So they could increase the expression of beta-gal and increase the yield of the A and the B proteins. So it's important to know about bacterial gene regulation in order to exploit this information. Okay. And so just to finish up on the subject of modern biotech, it's probably the most important discovery in pharmacy, along with antibiotics and vaccines, I think, that was made in the 20th century. And in 1980, the whole industry changed. You can see that once we can actually clone and express something in E. coli, it's a new way to make drugs. It's safer. We can improve them by changing this primary structure, and it's now used in vaccine production. Many drugs are produced by this methodology. We can use it in antibody technology as well. Some of the same principles are used. Okay. So I spent a bit longer on that than I hoped. What I'm going to do now is make a quick start on regulation of gene expression, and to just introduce to you the concepts of gene expression and why they're important to understand. And we could go through this pretty quickly because this is a bit of revision for you on the cell biology of the molecular biology of the cell. So here you have a bacterial cell. Here's the pointer. Sorry. Pointer. Here's the bacterial cell. It has DNA. This DNA is replicated in order to produce copies, which can allow for bacterial division. But also, the function of DNA is to make proteins and to encode proteins in the cell. So you know this from your previous lectures. This is the process of transcription, which makes RNA, and the process of translation, which makes proteins in the cell. This can be replicated, this can be represented like this. This slide, same sort of thing. DNA gives rise to RNA, slightly different structure. It's a single-stranded structure, and then this is translated to give proteins in the cell. What we'll see in this lecture and then the next lecture is that this is a very energy-sapping process. It uses a lot of energy to make RNA and then a lot of energy to make a protein in the cell. So the thing about gene expression is if you don't need this protein, then you shouldn't make it. You shouldn't go through all this process. And this is the whole point of regulating gene expression. If you don't need this protein, maybe you don't need the cell to make a certain protein, then if you can stop transcription, it will stop translation and you won't make the protein and you'll save a lot of energy in the cell. So we know, for instance, that, we know, for instance, that prokaryotes and eukaryotes are very, I'm just wondering whether to stop this here. It still leaves us with quite a lot to do next week. But I think what I'm going to do, I'm going to stop the lecture here, okay, with this concept. I'm going to finish with this concept. This is the concept that I want to discuss, and that's that the process of making a protein is it takes a lot of energy in the cell, okay? So the cell needs to use a lot of energy to make a protein. So what if the cell can regulate transcription so that when it doesn't need a protein, it stops transcription, stops everything happening, and it saves a lot of energy. And this is the process of regulating gene expression. And I may revise my slides a bit for next week because we've got an hour to describe this process of regulating expression in the cell. Okay, so what I'm going to do is I'm going to open up the chat. And if anybody has any questions, you have a couple of minutes to ask if anything is unclear. So what I've done today is I've described how insulin is made by the pharmaceutical industry, and using the history of genetic engineering to describe it's your industry, it's your biopharmaceutical industry is all based on that technology that was developed in the 1960s and 1970s, all leading up to the use and generation of insulin, which can now be made by these very modern approaches. And hopefully when you study the slides, you'll see, yes, I understand how to clone a piece of DNA into a cell, and then take that DNA, put it into a plasmid. That plasmid can express the protein. And if that protein is the A chain of insulin, I can then mix it with the B chain of insulin, put them together, and then we have insulin. And that is a wonder drug that can save the lives of people with diabetes. And we can use modern biotech. Okay. So I think that's enough for today. You have 3 or 4 minutes to get to your next lecture. And feel free to ask any questions. I can see maybe this is, hopefully it's clear. There are no questions. Thank you. Shakran, thank you very much. It's a pity I'm not there in person. I visited a few years ago, and it was a very nice experience. So it was very nice to meet some of your classmates who have now graduated. I met them a few years ago, and it's a pity I'm not there today. But I hope you were able to follow this lecture. Okay, thank you. Thank you. Thank you.